we want to grow this microorganism in the laboratory for the diagnostic purpose. So if we want to grow this in the laboratory, then we must understand what are the growth requirement of the microorganism, because we are providing all conditions. I repeat, we are providing all conditions which are there inside the body for the growth of the microorganism outside to the body or in the laboratory. So, which are these conditions? Question is, which are these conditions that we are providing to the microorganism outside to the body in the laboratory? So, microorganisms are easily grown in the laboratory or outside to the human body. So, I think this is clear in your mind. The purpose of study of this topic is very clear in our mind. I will show you some photographs as well, and we'll try to understand the nutritional requirement and growth of the bacteria in brief. So let us start with the basic nutritional requirement of the cell, living cell. So every organism, I am repeating, every organism finds all the substances required for the energy generation and the cellular biosynthesis in the environment. So everything is available in the environment for all living microorganism. The chemicals and elements of this environment utilized for the bacterial growth referred to as a nutrient. So this is a very simple definition of nutrients. Somebody may ask, what is the definition of nutrients? So the chemical and the element of this environment utilized for the bacterial growth. So this is referred to as a nutrient. Now, many bacteria can be grown in culture media. Now, what is the meaning of culture media? So yesterday, this was, I think, discussed in yesterday's lecture, or probably Chaitali mentioned regarding the culture media. I will also briefly introduce the culture media today, and there will be a separate separate discussion or session on the culture media, and there will be a practical on the culture media. So when you join the college again, we will show you all the culture media plates. Now let us move. Minimum nutritional requirement for the growth and multiplication of bacteria includes sources of CHON. So everybody knows these are the minimum requirements. CHON are the minimum requirements for the nutritional requirement for the growth and multiplication of bacteria. Now let us talk about the bacterial vitamins. Now, some fastidious bacteria. Fastidious means they are not easily grown in the laboratory. For them, now when I use word they are grown in the laboratory, that means we are using media or medium to grow them. So some microorganisms, some bacterial microorganisms do not grow in the laboratory. For them, some special culture media are required. And special culture media means what? They are enriched media, or they are some specific culture media. So in which we are adding some substances to make them efficient to grow this microorganism, fastidious microorganism, in the lab. Otherwise, they will not grow. Some microorganisms easily grow in the laboratory condition in a simple culture media. For them, basic culture media are enough. But for some microorganisms, we require to add some substances or some bacterial vitamins or some chemicals in the culture media plates to grow them. So there are few examples of vitamins along with the name of the bacteria are given in this chart. Biotin, Leuconostoc species, B12, lactobacillus species. Folic acid is required to grow Enterococcus faecalis. Now, Enterococcus faecalis is a pathogenic organism causing various infections in the patient. Then pantothenic acid, Morganella morgani, again like Enterococcus faecalis, Morganella morgani is a pathogenic organism causing various infections. Then vitamin B6, lactobacillus species, niacin, Brucella abortus, again Brucella abortus and Haemophilus influenza. These are the pathogenic organism causing infections in the patients. And riboflavin, vitamin B2, that is required to grow the Bacillus anthracis in the lab. This organism is responsible for disease anthrax. So I've given few examples of the substances along with the microorganisms. Now, what is the meaning of artificial culture media? Now, word itself is explanatory. Artificial culture media. Are you getting? Anyone can answer whatever I am saying. Are you able to understand? Write a message in the chat box. Okay, fine. So let us try to understand the meaning of artificial culture media. Now, why we are using artificial culture media? In the beginning of this presentation or session, I said our purpose of this session is to understand the growth requirement or nutritional requirement of the microorganism. To know this, because we want to grow them in a laboratory. Why we want to grow them? We want to grow them for the diagnosis. So we are using artificial culture media in the lab. Now, it is understood when we are using artificially prepared culture media, that means all substances, those substances available in the human body as a nutrients are must be available in the culture media plates. Otherwise, microorganisms do not grow. So we are providing all the nutritions, all nutritional substances which are available in the human body, they are providing artificially. So it provides sources of carbon, energy, and nitrogen in the form of available carbohydrates and amino acids. This is understood. Several media provides inorganic salts or particular growth factors. So these substances are available in the human body. So we are adding these substances and making a culture media. So when you are preparing a recipe for the culture media, these substances must be added. Now, there are two varieties of culture media: routine laboratory media and specialized media. Now, what is the meaning of routine laboratory media? To obtain suitable growth, the artificial culture media should provide nutrients and pH. Now, remember, we are also providing a similar condition outside to the human body. Now, which are these similar conditions? Say these similar conditions, like say we are providing on nutrients, we are also adjusting pH. Human body pH or tissue pH or blood pH is around 7. So most of the pathogenic microorganism grows at this pH. I am saying most of the pathogenic microorganism or organism causing human infections grows at around pH of 7, with few exceptions. Say like Vibrio cholerae grows at alkaline pH and fungi grows at acidic pH. Otherwise, most of the pathogenic microorganism or organism causing human infections grows at pH 7. So we are adjusting the culture media plate pH 7. Many of the nutrients supplied by the aqueous extract of meat and peptone. Now remember, we are exclusively using meat extract because meat provide most of the substances or most of the nutrients which are required for the growth of the microorganism, or you may say these nutrients are available in the human body. Now, there are various classifications of the culture media. Now, let us try to understand few of them, not all. So based on consistency, or you may say based on the phase, there are three types of culture media. Liquid culture media means the phase of this culture media is liquid. Semi-solid media means this media are not liquid and not solid. So they are jelly-like culture media or gel-like culture media, and solid media where we are adding some substance like agar to make them solid. So we are adding agar in the liquid culture media. So this agar makes this culture media plate solid. While semi-solid agar, we are adding agar but in low concentration. So the agar is an inert substance, physiologically inert substance, which do not add or decreases the nutrient or nutrition, but it only makes the culture media solid. So this is a difference in the consistency. There are purposes for a different consistencies culture media that we will see later on, why we are using a liquid media and why we are using semi-solid media and why we are using solid media for particular microorganisms or for particular biochemical taste that we will discuss later on. Now, second classification is based on the growth requirement of the microorganism. So routine laboratory culture media are able to grow the routine pathogens. I'm repeating this statement. Routine laboratory media or culture media plates are able to grow the routine pathogenic microorganism. Like simple or basal media. Enriched media means we are adding some substances like blood or some blood or egg or serum, which increases the efficiency of the basal media, that is known as an enriched media, or we heat the blood agar. So once we heat a blood agar, RBCs present in the blood are broken and some bacterial vitamins are released from the RBCs. Some factors like X and V factors are released from the RBCs and provide nutrition to the particular pathogenic microorganism. So enriched media, enrichment broth. Now broth, when it is written broth, it means its consistency is liquid. So enrichment broth select out particular organism from the mixture of organism. So this enrichment media allows the growth of the pathogenic microorganism. In other words, this enrichment broth increases or it gives support for the multiplication of the pathogenic microorganism. Selective media word itself is explanatory. When you want to select some particular microorganism, some substances are added in the culture media. Selectively it grows the micro. Say for example, McConkey agar medium is selectively grows the Enterobacteriaceae group of organisms. Or say some antibiotics are added in Saborodextrose agar, which inhibit the growth of bacterial microorganism. In other words, they favor the growth of the fungus. So selective media, where some substances are added, deliberately added to allow the growth of pathogenic microorganism. So this is known as selective media. Differential media, which differentiate the two groups of organism. Yesterday we have discussed regarding the differential staining technique, where differential word itself explanatory, where if we want to differentiate two groups of organisms, we are using differential staining, or if we want to differentiate two groups of microorganisms, we are using differential media. So we'll discuss later on in the session of culture media. Transport media, I will explain all the culture media when you come again to the laboratory or when you join the college. I will show you and describe all the culture media in front of you. Transport media is again explanatory word, where say suppose we want to transport some microorganism from one place to other place, or say suppose we want to transport some sample from one place to other place. Say, for example, VR medium or Amis transport medium or Carry-Blair medium. These are the examples of the transport media. Anaerobic media, again, explanatory, where say if you want to grow the anaerobic microorganism, then these culture media are used. Defined or synthetic media, simple synthetic media and complex synthetic media. Later on, we'll discuss regarding this. We are not using in the lab routinely, but this is something regarding the classification of culture media. Now, this is a ingredients of the culture media. We are not going to discuss the recipe of the culture media. Now, suppose these are the ingredient: water, agar, peptone, casein, meat, yeast, malt, blood serum. All substances, we know all substances contain the nutrients either in the form of carbohydrate, protein, or amino acid, lipids. Everything is there in these substances. So the recipe is simple, where there are say either you purchase all these substances from the market and weigh it and then mix it and then prepare a culture media. This is one way. Second way is dehydrated powder form is purchased from the market, which contain all these substances, and it is dissolved in a laboratory and culture media plates are prepared. This is the second method. Third method, laboratory can purchase culture media plates directly from the market. So there are different purposes. Say, for example, first method is cumbersome and there is no guarantee of the quality. When you purchase all the different ingredient and mix it and prepare a media, that is not a good idea. Second method is where you purchase a powder from the market and dissolve it and prepare a culture media. This is reasonably a good idea, because the dehydrated powder form having those companies who are selling this dehydrated form are taking responsibility of the quality of this powder. So this is an advantage, and you don't have to weigh all the substances. You don't have to purchase all the substances from the market. Just purchase a powder which contain all these substances and dissolve it and prepare a culture media. Or third method is very simple, where you purchase a ready-made culture media plates. Where third method is where when you purchase the culture media plates, then it is a costly affair, because when you use a second method, one culture media plate costing around 2 to 3 rupees, where when you purchase culture media plate, it is costing around 30 to 40 rupees per plate. So this is a difference, but there are other things also. In our lab, we are using third method. We are purchasing the culture media plates directly. Now you see this is a culture media plate. Now this is a photograph where it's showing the growth of the pink color organism, or pink color colonies are seen. Okay, say colony means it is a it contains the similar type of microorganism, pure. Say when we say colony, it is always pure. There is no contaminant in particular colony. Say suppose for example, this is a colony of Escherichia coli. That means all cells in this colony are of Escherichia coli. There is no question of the contamination. When I say this is a doctor's colony, it means all residents of this colony are doctors. So where other category is not allowed, or say contaminants are not allowed. Only doctors are staying in the colony. That means it is a doctor's colony. A similar way, when I say these are the colonies of Escherichia coli, that means one colony containing all cells of Escherichia coli, where Klebsiella pneumoniae is not allowed. In doctor's colony, engineers are not allowed like that. Say this is for the comparison. This is on the MacConkey agar medium. Escherichia coli shows the growth, and colonies are of pink color because it ferments the lactose, and pH of the medium is reduced because of lactose fermentation, and color of the colony is changed. While Salmonella shows the colorless colony on the medium. So if one is lactose fermenting colony, other is non-lactose fermenting colony. So this is on the differential culture media plates, the color differentiation. This is a blood agar plate. Now, in the nutrient agar medium, blood is added. Now we are not adding human blood, we are adding animal's blood in this, like say sheep blood or horse's blood, because human blood containing substances which may kills the microorganism. So this is not a good idea to add human blood in the culture media plate where we want to grow human pathogens, because when you add human blood, then human pathogen will not grow, because human blood may contain the substances against the human pathogen. So we are using sheep blood. So this is a sheep blood agar where 5% concentration of blood is added in the nutrient agar broth, and then media is prepared. These are the colonies grown on the sheep blood agar, where the color of the colony, now they're going to detail of that, but what is the size of colony, what is the shape of the colony, what is the color of the colony on the blood agar, whether there is a hemolysis seen or not. So such points are noted down for the identification, primary identification of the microorganism. I am facing little bit problems in the moving of the slides. So this is a next slide where this is a clade agar medium on which the colonies of Pseudomonas are grown. Now we'll discuss later on what are the characteristics of Pseudomonas on the clade agar medium, and what are the characteristics of the clade agar medium. This is another plate where organisms are grown, colonies are seen on the culture media plate. Now the purpose of showing these photographs at this moment is to make you understand we are using to grow the microorganism in the laboratory. Purpose of growing this microorganism in the laboratory is to identify them. So this is our purpose, and once we are able to identify them, then diagnosis is clear, where say what type of infection is there in the patient, and then according to the infection, antibiotics are given to the patient. Now let us move to the another objective. What are the nutritional requirement of the cells? Now this is a classification based on the nutritional requirement of the classified, or the organisms are classified into 4 categories: phototrophs, chemotrophs, autotrophs, and heterotrophs. Now all are self-explanatory. Autotrophs are organisms that uses carbon dioxide as a sole source of carbon for the growth. Heterotrophs utilizes preformed organic compound. So remember, out of these 4, most of the pathogenic organism, when I say pathogenic organism means infection, the organism causing infection to the human host, are falling under the category of heterotrophs. So let us try to understand what is the meaning of growth. Are you getting whatever I am saying? Are you with me? All students are with me? Okay. Thank you. Meaning of growth is increase in the quantity of the cellular constituents. This is one meaning of growth. We know the meaning of growth in the humans, but let us try to understand a meaning of growth in the microorganism. Increase in the quantity of the cellular constituents. So it forms a new protoplasm from nutrients available in the environment. So एक नया protoplasm बन रहा है from the nutrients available in the environment. This is the meaning of growth. Increase in the cell mass and the number of ribosomes. Duplication of the bacterial chromosomes. synthesis of new cell wall and plasma membrane, and partitioning of the two chromosome, septum formation, and cell division. So this is a meaning of the growth, where one cell become two, two cell become four, four become eight, like that. So there is an exponential or logarithmic growth or logarithmic increase in the number of the cell. So this is a binary fission. So I'm not going into detail of this. All students knows regarding the binary fission. Okay. So we'll discuss later on various type of reproductive methods in the microorganism. But this is a simple binary fission where division occur and one cell become two. One is a parent cell, other is a daughter cell. Okay. Now, what is the meaning of growth rate and generation time? This is very important to understand. When we are growing microorganism in the laboratory, we must know what is a growth rate. At what rate this microorganism will grow. है ना, patient आगे पूछेंगे कब report मिलेगी हमारी. So if we do not know the growth rate and generation time, we are not able to answer this question. है ना? There is a concept of turnaround time where we want to convey the timings to the patients or the clients, where clients are asking at what time my report will come. So on the basis of this, we can tell the clients where your report will come after 48 hours, or your report will come after 72 hours, or your report will come after 1 hour or 2 hours or 5 hours. Okay. The cell divide at a constant rate depending upon the composition of the growth medium. So this all depend upon the composition of the growth medium or availability of the nutrients. If nutrients are not available, then their growth is not possible. Okay. And second is condition of the incubation. So there are two factors. Remember, the growth is constant, but it depends upon the two important factors. And one is availability of nutrients in the nearby environment, and second is incubation condition. Now, the rate of exponential growth of the bacterial culture is expressed as a generation time, also as a population doubling time. So we know the meaning of generation time in the human being. Now, what is the generation time in the microorganism, which is definitely different from the generation time in the humans? Let us try to understand the different generation time of the commonly isolated microorganism in the labs. The generation time of Escherichia coli in the laboratory, 15 to 20 minutes, 12 to 24 hours in the intestinal tract. So remember, generation time for this commonly isolated organism in the lab, say 15 to 20 minutes. While in the intestine, one become two. For most known bacteria, generation time ranges from 15 minutes to 1 hour. So this is the answer of the question. For most commonly isolated bacteria, generation time ranges from 15 minutes to 1 hour. There are a few exceptions like Mycobacterium tuberculosis and Treponema pallidum, where generation time is very long. It is in days. So that's why when we in previous time where Mycobacteria or tuberculosis was suspected, samples were taken and inoculated on the culture media, and we give a turnaround time of 2 months to the patient. 2 months. Patient को बताते थे 2 महीने के बाद report मिलेगी after 2 months because of long generation time. Okay. But ask patient, because there is a automation and there is a advanced technology where we can ask the patient generation time turnaround time 2 महीने से कम होके 1 to 2 hours हो गया अभी, है ना? So such advancement is happened. So Mycobacterium tuberculosis and Treponema pallidum have a long generation time. Now, second question is how to measure the growth. This is important because this is related to the prognosis of the patient or whether treatment is effective or not. So this is indirectly related to that. So how to measure the growth? So the very commonest method is physical measurement. of dry weight or volume of the cell after the centrifugation. So this is a very simple method and time-tested method where we can do a centrifugal, we can give a centrifugal force at the top of the test tube, and at the bottom you will find the sediments, and you weigh the sediments. So this is possible. Second is chemical measurement of some chemical component of the cell, such as N or total protein or total DNA content. We can measure the growth, or measurement of chemical activity such as rate of oxygen production or consumption or carbon dioxide production or consumption. So we can do like this. I will take little bit more time because I want to finish this topic today. So you are at the home, so no problem, I think. Measurement of the growth. This is another method where turbidity or optical density of a suspension of cell is directly related to the cell mass or cell number. So यहाँ पे हम देखेंगे कि turbidity कितनी हुई. अगर turbidity, by the measurement of this turbidity or optical density, one can say, and the number of the cells are extrapolated based on the turbidity or optical density. And one can give a number 10 raised to the power of 2 or 10 raised to the power of 3 or 10 raised to. So densitometers are available, so which directly convert the turbidity into the number of the microorganism grown. So this method we are using in the laboratory to measure the turbidity by using densitometer or measure the density. So this is a one method to measure the growth. Method of measurement for cell number, one can do a direct microscopic examination where we are also counting the number of the microorganism, number of the WBCs, and number of the epithelial cell present. Okay, so this is by the direct microscopic method. There are examples where we wanted to count the number of the WBC and the epithelial cells and the presence of microorganism to for the further identification. Then electronic counting chambers are available, or indirect viable cell count, also called plate counting. Now plate counting is another method where in the urine culture we are doing plate counting, or in the water sampling we are using this method. Okay. Now, before we move further, I'll give you one example of the importance of the counting of the microorganism, where in urine culture there is a hypothesis. Now, urine culture is taken in case of suspected urinary tract infection. So, where we are counting the number of organisms present per ml of urine. This is important to fulfill the requirement of the hypothesis. Now, hypothesis is like this, where say if number of microorganism or colony forming units, say 10 raised to the power of 3 per ml of urine, then it is not infection, it is contamination. Say second statement of this hypothesis is like this: when number of colony forming units are say between 10 raised to the power of 4 to 10 raised to the power of 5, then it is possibly infection or possibly contamination. So it is in between or indeterminate, where you have to see the patient's condition or some factors or host factors. And in third statement, if number of colony forming units are more than 10 raised to the power of 5, then it is probably infection along with some conditions. So this is the importance, where there is another example of the bronchoalveolar lavage or say respiratory infection. Here we are counting the number of the colony forming units. So because we want to fulfill the hypothesis and contamination. So this is importance of counting the number. There is another slide which shows the total number or total count and viable count. So there are various methods. I'm skipping this slide. You prepare it from the book. Now, this is a important uh, curve that we want to understand. Now you try, say there is an experiment over here. Let us try to understand this experiment. Now, this is a graph of time versus the number of cell. Now, how this graph is prepared. Now, at the 0 point, you try to understand at the 0 point, if you inoculate the microorganism in a liquid culture media, there will be a lag phase. For some period of time, there will be no growth. This is time for the adjustment. Now, any or every living thing takes a time for adjustment. In a newer condition, all living things take some time for the adjustment. Now, once they adjusted in a newer environment, all nutrients are available, incubation conditions are favorable, other conditions, gaseous conditions or availability of gases are favorable, then there will be a growth. And this growth is, say, known as an exponential phase or log phase. Then once, now what happens? This is not forever. When at certain period of time, after certain period of time, there will be a stationary phase. In the stationary phase, newer generating cells and dying cells become equal. Total number remains the same, but the number of dying cells and number of newly generating cells are same. So there is no reduction in the total number. This is known as a stationary phase. At one point of time, cells are dying because of there are some reasons for this. Say one reason is exhaustion of the nutrients, because nutrients are not plenty. There is a limit of the nutrients. So at one point of time, nutrients are exhausted and there is a decline phase or death phase for most of the growing bacterial microorganism, and there is a decline phase. When there is a decline or death phase, number of the cells are reduced. Another reason in the human body, say there is an immune system, where immune system tries to kill the microorganism. So there is a death phase in the human also. This is in we understand each and every phase, some theoretical aspect. Lag phase, I'm reading the theory. Whatever I said, immediately after the inoculation of the cell into the fresh medium, population remains temporarily unchanged. There is no apparent cell division occurring. Cell may be growing in volume or mass, synthesizing enzyme, proteins and RNA, and increasing in the metabolic activity. Everything is happening, but they are not multiplying. So this is in the lag phase. Exponential phase, where pattern of the balanced growth, where all the cells are dividing regularly by binary fission. Stationary phase, exponential growth cannot be continued forever in a batch culture outside to the human body. So in vitro, some cells are dying and equal number of cells are dividing. So population growth is limited. Now the number remains same, and population growth is limited by the exhaustion of the nutrients and accumulation of the inhibitory metabolites or the end product. And finally, death phase, the viable cell population decline. During the death phase, number of the viable cell decreases exponentially. So I think all students understood this growth curve. Now, what is the application of this growth curve? I'll show you the application. Now you try to understand this: bacterial divide, bacterial death, total count, viable count, and spatial features, lag phase, log phase, stationary phase. So you make up a chart like this and try to understand. In the prelims exams, we have asked this question to the students as a 5-mark short note, growth curve. This will be a direct question, because we are also asking some direct questions where you don't have to think, just write it. So this is one of it. Now this is the application of the growth that we are doing. We are using this in the laboratory. Now on the right side, 2 bottles are seen. These bottles are the culture media bottles, where we are inoculating blood or the body fluid. When we are suspecting septicemia or infection of blood or infection of central nervous system or infection of the joints, where we are collecting fluid, or infection of the pleural cavity. So we are collecting fluid, body fluid or blood, and inoculating these samples in these bottles. Now, type of medium is broth, and which is also known as an enrichment broth. Okay, so try to understand, type of medium is enrichment broth, and we are using it for the inoculation of blood sample and the body fluids. Later on, I will make you understand what is the concept of this inoculation and what is the concept of growth. But here in this media, in short, in this medium, there are two phases. One is liquid phase, other is solid phase. At the bottom, there is a solid phase, and there is a thin membrane between liquid and the solid phase. Now, when blood sample or any sample is inoculated in this bottle, if microorganisms are present in this sample, they grows. Okay, after the lag phase, once they multiplies, carbon dioxide. This carbon dioxide allowed to fuse into the solid media, because thin membrane is there between the solid and liquid media. So this thin membrane allows only carbon dioxide, no other gases are allowed, only carbon dioxide allowed. Now, once this carbon dioxide fuses into the solid media at the bottom, the pH of this bottom media is reduced, because carbon dioxide reduces the pH. So pH is reduced, and there is substance, pH indicator is added in the solid medium. So color of this bottom is changed. Now, once color of this bottom is changed, which is identified by the calorimetric reading. So on the left side, there is a machine, BacT-Alert. Now, this machine we have, and this machine identify every 10 minutes, this machine take a reading, and there is graph generated on the screen. Okay, there is a computer screen attached to this. So graph generated on the screen, and this graph showing the number of the microorganism or bacterial microorganism grown in this bottle. Now, at one point of time, this machine gives a signal when it reaches to the threshold. Once this machine gives a signal, we remove this bottle and inoculate this content of this bottle on the solid culture media plate, and the colonies are grown. So this is a method for the identification of the presence of the microorganism in this bottle. It means once presence of microorganism in this bottle, that means presence of microorganism in the body fluid or in the blood. So I think this is a application of the concept in the laboratory, direct application. Now, such type of graph is generated on the computer screen, where it shows now total number of colony forming units are calculated by the machine. Now, let us move to the last and final objective of this session, which are the other requirements for the microbial growth. One is now there are gaseous requirement, like oxygen is universal component of the cell, provided in large amount by H2O. Now, type of respiration you already learned in the first MBBS. Let us move fast. Obligate anaerobes that require O2 for the growth. They use oxygen as a final electron acceptor in the aerobic respiration. So you must have learned about the Krebs cycle and other cycles in biochemistry. So we are not going into that cycles. Obligate anaerobes do not need or use oxygen. Facultative anaerobes, organisms that can switch between aerobic and anaerobic type of metabolism. So remember, most of the pathogenic microorganisms are falling under the category of facultative anaerobic organism. So they can switch between aerobic and anaerobic types of metabolism, with few exceptions. There are few organisms which are strict aerobe and few organisms which are strict anaerobe. Otherwise, most of the pathogenic microorganisms falling under the category of facultative anaerobic organism. Then there are aerotolerant organisms, anaerobe bacteria, which are exclusively anaerobic fermentative type of metabolism, insensitive to the presence of oxygen, and they live by fermentation alone, whether or not O2 is present in their environment or not. So this is aerotolerant anaerobic organism. They are few in number, when we are talking about the pathogens. Now this is a chart that you prepare with examples. Obligate aerobes can grow only in the presence of oxygen. Pseudomonas, Mycobacterium, tuberculosis, Bacillus, Brucella, and Nocardia. These are the few examples of the microorganism. They are obligate aerobes. So they only grows in the presence of oxygen. Facultative anaerobes, most of the pathogenic organisms are falling under this category. They can switch between aerobic and anaerobic. But usually we are growing this microorganism in an aerobic condition. Facultative aerobes, they are anaerobes. They can also grow aerobically. Example is Lactobacillus. Then there are other examples: microaerophilic bacteria, Campylobacter and Helicobacter, obligate anaerobes. Clostridium is the best example of obligate anaerobe. Once they are exposed to the oxygen, they are killed. And aerotolerant anaerobes, Clostridium histolyticum. So these are few examples of the microorganism or pathogenic microorganism. There are many, but I am only talking about the pathogenic microorganism which are strict anaerobic organism. For them, you require to provide anaerobic environment. Now, finally, there is something regarding the pH. Minimum pH below which organism cannot grow, maximum pH above which organism cannot grow, and optimum pH. So we want optimum pH at which organism grows best. So we are adjusting the culture media plates pH or culture broth pH optimum, so which allows the growth of the microorganism. So there are 3.7. There are 3 point at below 7, at 7, and above 7. So but most of the pathogenic microorganisms grow at pH 7, around 7. So we are adjusting the pH 7.2 in most of the culture media plates or the broths. Then temperature, psychrophilic, ability to grow below 10 degree, mesophilic organism with an optimum temperature near 37. So remember, most of the pathogenic microorganism grows between 25 to 40 degree temperature. This is a range. And optimum temperature is 37. This is understood or logical, where human pathogen grows based at 37 degree temperature. So we are adjusting the temperature 37 degree. There are some thermophiles grows like say optimum temperature between 45 to 70, extreme thermophiles. But remember, these organisms mostly they are saprophytic organism. Pathogenic organism having a temperature range between 25 to 40 degree. Then water is an essential ingredient of the bacterial protoplasm. So drying is lethal. Drying is lethal to the cell. So this is also important to understand when we want to kill them, because we are doing two business. One is we are growing them in a laboratory for the diagnosis, and we are killing them to make surfaces clean or sterile. So these are the two businesses of the microbiologist: grow and kill. So killing is again very important to understand. So drying is lethal to the cell. Later on we will also discuss on the how to kill the microorganism in a chapter of sterilization and disinfection. Light, bacteria grows well in the dark. So we know microorganism grows best in the dark, with few exceptions where they only grow in the presence of light. But most of the pathogenic microorganism grows well in the dark. They are sensitive to ultraviolet light and other radiation. So this is also important to understand. So because I said we are in two businesses: grow and growth the organism and kill them. So say suppose we want to kill them, we are using ultraviolet light. Sunlight is also having some killing effect on the microorganism. Osmotic effect, bacteria more tolerant to osmotic variation. Plasmolysis, sudden exposure to the hypertonic. This is known to everyone. What is plasmolysis and plasmopsis? So this is regarding the osmotic effect. Mechanical and sonic stress, cell wall may be ruptured by the grinding or vigorous shaking with the glass beads. This is also important because in some tests we are uh, breaking the cell walls because we want the nucleus of the cell for some test. So in the situations where we are rupturing the cell by doing vigorous shaking or grinding them, or disintegrated by the exposure to the ultrasonic vibrations. So ultrasonic vibrations kills the microorganism. So this is end of the chapter or discussion. You read this chapter from the textbook today only. There are a few questions: how bacteria grow and multiply, and list the salient features of the bacterial growth curve. So this may be a 10 marks question, but nowadays we are not asking such question as a 10-mark question. Uh, we are giving the patient profile to the students in a 10-mark questions. We are not asking us similar type of question or such simple questions, direct question to the students. We are asking the question where you have to think or analyze the situation. But anyway, this may be asked as a 5-mark short note. Second question, discuss salient aspect of the nutrition, gaseous requirement, temperature requirement, and other physiological requirements of bacteria for the growth. So this is another question, direct question, which falls under the category of 35%. We are asking 35% direct question and 65% questions where you are required to think something. Thank you very much.